Hello, everyone. Let's get started. My name is Alan Hudson from the Immuno-Oncology Translational Network Data Management Resource Center. I also serve as Chair of Biostatistics and Bioinformatics at Roswell Park Comprehensive Cancer Center. Before we start the seminar, I just have a few bookkeeping items to go over. Questions will be responded to at the end of the presentation. Please enter your questions in the Zoom question and answer box. The audience can prioritize the order of the questions by clicking on the thumbs up button. Closed captioning is available. The closed captioning for today's webinar can be accessed by clicking on the live transcript option in the Zoom menu at the bottom of your screen. Instructions are also provided in the chat box. If you're having technical issues, you can make a comment in the Zoom chat box. With that, I would like to turn the proceedings over to our moderator, Dr. Paul Sondel, who is the Reed and Carol Lee Walker Professor of Pediatric Oncology in the Department of Human Oncology at the University of Wisconsin School of Medicine and Public Health. Dr. Sondel is a world-renowned leader with respect to investigating immunotherapy approaches towards combating pediatric cancers. Dr. Sondel.
Thanks very much, Dr. Hudson. Welcome, everybody. It's really a pleasure and privilege to introduce my friend and colleague, Dr. Paul Sorensen, a colleague from the Pediatric Immunotherapy Discovery and Development Network and from the Pediatric Cancer Dream Team. Paul is a native of Denmark. He is the Joe Hall Chair Professor of Pathology at the University of British Columbia in Vancouver, Canada, and is a distinguished scientist at the British Columbia Cancer Research Center. He is an internationally renowned expert in messenger RNA translation and the use of proteomic strategies to characterize cell stress pathways and the potential relevance of these pathways to metastatic capacity. His group has discovered many novel genetic alterations in childhood cancer, including the EWS-ERG fusion in Ewing sarcoma and the ETV6 NTRK3 fusion in infantile sarcomas and secretory breast carcinoma. The first description of NTRK fusions as recurrent drivers in human tumors, now thought to occur in greater than 25 different human tumor types, leading to the development and worldwide approval of NTRK inhibitors for clinical use. He has won many national and international awards and is an inductee into the Royal Society of Canada and a member of the Order of British Columbia. I'm really looking forward to his talk. Professor Paul Sorensen.
Thank you so much, Paul. It's a pleasure to be introduced by a good friend and co-member of some of these initiatives that you mentioned. I just want to make sure we can see my slides, which I hope we can.
Yes.
And then I've actually learned how to hide some of the other prompts that are up there. So thanks again. And it's a real pleasure to be able to present some of our work to this illustrious network. And I thank you for the invitation. So we're going to talk today about our strategies for identifying new immunotherapy targets in high-risk childhood cancers, and how doing so not only gives you tools for immunotherapy, but also insights into interesting biology. So this actually started innocently enough, probably about 7 or 8 years ago, as part of the Stand Up to Cancer St. Baldrick's Foundation Pediatric Dream Team that Paul and I are members of. In those days, we wanted to use differential gene expression, comparing pediatric cancer tumor to normal tissue, and identify candidate targets based on selection of genes predicted to encode cell surface epitopes. We then set up a PathCore in Vancouver to score tissue microarrays for high expression in tumors and low normal tissue staining. And then the idea was to come up with antibody drug conjugates or antibodies to engage the immune system, or make CAR Ts and bispecifics to select agents for clinical development. But along the way, we learned a few things. And one was that proteomics is probably a better way to find surface targets than gene expression profiles. And the other thing we learned is that it's really important to choose targets that confer some kind of survival or proliferative advantage to tumor cells. And so what I'm going to tell you about today is really a team effort between our group in Vancouver and the names you see here, and the group at Children's Hospital of Philadelphia with John Maris and others, as you see here, and some key collaborators such as Mitko Dimitrov, Rimas Arentas, and Crystal Mackel. So the approach we took was to start the whole process by looking at patient-derived xenografts and do proteomics on these entities. And I've listed them out here. And the reason for that is that there exists a set of PDXs from the PPTC run by the group at in San Antonio, and there's all sorts of drug screening and RNA-seq available on these samples. So we thought this would be a good place to start. And we're not going to go through all of this, but the ones in red are the ones that we're studying in Vancouver: medulloblastoma, Ewing sarcoma, osteosarcoma, rhabdoid tumor, and Dicer1 mutant tumors. And John's group is doing the rest. So the work plan that we've established collectively between the two groups is to really compare the PDX surface proteomes, and I'll tell you in a minute how we do that, to a 12 cancer cell line mix, and I'll explain that. So basically, we came up with a very complicated multiple centrifugation and sucrose gradient mechanism for purifying surface enriched proteins and then loading them onto our fancy machines for proteomics. At the same time, we take a small amount of the lysates and we do whole proteomics on the same sample, so that we can really compare the whole proteomes with the surface proteins. So just a little bit about the super mix that we compare the PDXs to. This is a panel of 12 cell lines that, for our purposes are unrelated to pediatric cancer, to use as a quantification reference as a comparator. We also do something called tandem mass tag, or TMT isobaric technology, which allows us for really excellent mass spec quantification, and the plexes also contain pooled internal standards and this cell line mix that I mentioned to you. So the idea of this is to really get good quantitative information on the surface proteins that we're looking for. And if people are interested in the details of the method, I'm happy to talk sort of online or in the discussion period. So like I said, one of the tumor types that we're interested in this context is Ewing sarcoma. And I think everyone here knows that Ewing sarcoma is a bad actor among childhood sarcomas. It's the second most common bone tumor in kids after osteosarcoma. Very, very undifferentiated appearance with these small round blue cells. And as everyone knows, metastatic disease has a really dismal prognosis in this disease compared to localized. The big issue around looking for genetic drivers, of course, is that these tumors are among the lowest mutational burden containing tumors that exist. And as you all also probably know, they're largely driven by two gene fusions that fuse parts of the EWS RNA binding protein to either FLY1 or ERG, ETS family transcription factors. And so this generates in probably close to 99%, this says 95, but it's probably close to 99% EWS FLY1 and EWS ERG oncoproteins, which function as chimeric transcription factors. And we will come back to that in a little bit. So back to the surface enrichment. These are some of the raw data that came off our recent experiments. This is sort of our down-selected list of genes that or proteins that we're interested in. We look for fold change overall versus fold change on the surface. We also factor in a program called Surface Genie, which is an algorithm to identify surface proteins. We come in and we look at the percentage of the PDXs or available cell lines that have these particular proteins expressed. We factor in whether or not some of the proteins are known to be EWS FLY1 targets. And then we also look for transmembrane regions, which is another parameter of this being a surface protein. Then, of course, probably the most important thing from a therapeutic point of view, we want to find surface proteins that are not expressed at a high level in normal tissue. And so what we're looking for are ones that where these boxes are either all gray or all white. And from all of this, In Vancouver, we're currently looking at these proteins, and we'll go through them in a minute. You may have seen the annual meeting the other day where Brian Mooney talked a little bit about CDH11, so I won't talk about that today. So these are the 4 surface proteins other than CDH11 that we're looking at in our lab. I'm going to spend a little bit of time on a couple of these, a little bit more time on STEAP1, and then spend some time talking about ALA1RAP towards the end. So starting with ENPP1, I'm not going to say a whole lot about this. This is a very interesting surface protein that has a pretty good expression profile in Ewing sarcoma. It's not unique to Ewing sarcoma. This is the cancer cell line encyclopedia. We have lots of other data, and I'm not going to show you, but there's reasonable expression across these red dots, which are Ewing sarcomas, compared to other cancer cell lines. And just as a quick snippet, ENPP1 is a very interesting protein. It's involved in the cGAS STING pathway in that cytosolic double-strand DNA and probably also RNA activates the cGAS pathway to secrete cGAMP, which can then activate immune cells. But ENPP1 is a phosphodiesterase that can actually block this process. So it's sort of an anti-immune response protein. So we've got some antibodies to this protein, and we're moving ahead with interesting studies around that. This is an interesting protein as well, ATP11C, which is something called a phospholipid flippase. Here we actually see enhanced expression in Ewing sarcoma compared to other tumor types. And what we're excited about this is that this may actually be a direct EWS-FLI1 and ERG target, but I don't really have time to tell you about this today. Just want to mention that the function of this protein is super interesting. What it does is it flips the phospholipids, phosphatidylserine and phosphatidylethanolamine in the surface membrane, taking the phospholipid head group and flipping it to the inside of the plasma membrane. And why is that important? Because otherwise, these phospholipid head groups would be the target of macrophage eat me signaling. And so we're super interested in this as a way for tumor cells to avoid clearance by immune cells. So STEAP1 is a protein we've actually been looking at for a long time, even before the proteomics. This was highlighted years ago to be an upregulated protein in Ewing sarcoma. This is work by Stefan Burdak in Munich. This is the structure. There's a fair bit of loops that are actually outside the cell. We've become interested in this, and I don't have time to tell you, but it does seem to be a Ewing sarcoma fusion target. Here we see nice surface staining in Ewing sarcoma and much less staining across the board in other tissue types, highly expressed in Ewing sarcoma compared to other pediatric tumors, and also expressed in prostate carcinoma, where I believe it was actually first identified. So it's kind of a general theme. We do try to link our surface protein discovery program to adult malignancies because of the obvious implications for connecting to pharma for therapeutic targeting if we can find adult indications. Anyways, these STEAP family proteins are really interesting. They share this coupled iron or copper reductase activity that you see here with an NADPH oxidase activity. And the idea of this is that it creates ions of either iron or copper that can then be taken up into the cell. Now, what's interesting about STEAP1, which is sitting here, is that it actually doesn't have an NADPH oxidase domain. And so we thought that was kind of strange. And so we did some interactomics and found that if we pull down STEAP1, we can also pull down this other family member called STEAP2. And so STEAP1 and STEAP2 are both, we believe, EWS-ETS target genes, EWS-FLI1 and EWS-ERG. They're both highly expressed and highly correlated. And we've been doing a lot of ChIP-seq, and it's pretty clear that they're both coordinately regulated by EWS-FLI1. I don't have time to tell you about that, but icely expressed at the protein level across many different Ewing sarcoma cell lines and tumors. So what do these proteins do? We've been studying this mainly in the context of STEAP1. We made knockout lines by CRISPR, And one of the interesting features we've seen right away is that the iron 3+ reduction rates are much lower in the knockout cells. And this actually correlates with a reduced total iron content in cells lacking STEAP1 expression. And we're doing the same thing with STEAP2, but we're a little behind on that. So what does this mean? So it turns out that this is very important for getting iron into the mitochondria. And you know that there are a lot of iron-dependent enzymes in the mitochondria. So here on the left, here's one of these enzymes called aconitase 2. And you can see here in the red and green bars that cells with STEAP1 knockout have a much reduced activity of this iron-dependent enzyme. There's also an assay called RPA, which is a fluorescent assay whereby iron in the mitochondria quenches this fluorescent readout, and so you get enhanced fluorescent activity in cells that lack mitochondrial iron. And so finally, this correlates very nicely with a reduced ATP content in STEAP1 null cells. So we really think that STEAP1 is regulating iron uptake into mitochondria and augmenting mitochondrial respiration and efficiency. We also have been looking at mitochondrial membrane potential. So this is very interesting because, as you know, proliferating tumor cells need a lot of iron to drive the TCA cycle and oxidative phosphorylation. So that's where we're going with this. Now, from a targeting perspective, Amgen, this is data that they presented at a recent ACR meeting, have actually taken the first step in doing this. We'd like to think that we're right behind, but they've actually made a bispecific targeting both STEAP1 and with a CD3 binder to attract T cells. And they have preclinical efficacy in a Ewing sarcoma cell line where this bispecific is actually reducing tumor growth when human T cells are injected. And we've actually also got a set of STEAP1 and now STEAP2 binders that we hope we can create sort of bispecifics that are targeting both sides of it. But that's a little bit early, so I'm not really going to talk any more about that. But we're very excited about this as a set of targets for Ewing sarcoma and potentially prostate carcinoma as well. So for the last part of the talk, I'm going to focus on IL-1RAP, which is one of our most interesting targets. And this really started when, with a long-standing interest in my lab, which is to understand why the metastatic process where tumor cells leave the primary tumor, circulate, and then set up residence at a secondary site. Why is this process so inefficient? And most of us in the field believe that this is because each step of the metastatic cascade has a set of stresses that can actually cull pre-metastatic tumor cells: hypoxia, oxidative stress, shear stresses, and many others. And we got into this some years ago by showing, for example, that HIF-1 alpha is translationally activated by this RNA binding protein called YB1 to promote sarcoma metastasis. So we think that hypoxia is a very important driver of metastasis if the tumor cells can mitigate the stresses of hypoxia. Several other groups, such as the group of Sean Morrison at UT Southwestern, have shown that oxidative stress is actually directly inhibitory to metastasis by melanoma cells. So there's this general interest in understanding how tumor cells dial down these stresses to enhance their metastatic capacity. And so over the last few years, we've become really interested in reactive oxygen species and redox, loss of redox homeostasis as a way to inhibit metastasis. And I think everyone here knows that reactive oxygen species can be generated by many different pathways in tumor cells: hypoxia, metabolic defects, oxidative stress, and various oncogenes. And tumor cells need to dial up these antioxidant pathways to really enhance their metastatic capacity. So I'm going to tell you a story today led by Haifeng Zhang in the lab with help from Alberto Bailedelli that focuses on this major antioxidant called glutathione. And just to remind you what glutathione is, it's a tripeptide containing gamma glutamate, cysteine, and glycine. And what's interesting about this is that the sources of these different amino acids that make up glutathione come from varied sources. So the glutamate comes from glutamine via the action of glutaminase. Serine is converted from glucose, and this is converted to glycine. And then finally, what we'll talk a fair bit about today, cysteine, is actually generated either from the diet through cysteine or de novo through methionine and serine. So I'll come back to that in a little bit. So I come back to this slide on the metastatic cascade. And so what Haifeng actually got interested in was this part of the process. How is it that tumor cells survive when detached from the extracellular matrix? You've all heard of anoicas, which is cell death due to loss of contact with the extracellular matrix or adjacent cells that normal cells, normal epithelial and mesenchymal cells undergo when they lose detachment. Well, tumor cells have found a way to actually suppress anoicas, and so they can survive in the circulation theoretically, and this is linked to metastatic capacity. So to study this process, what a lot of people in the field do is they grow 3D spheroids in low detachment plates. And so we did that with Ewing cells. And one of the first things we noticed was that moving from 2D to 3D, Ewing sarcoma cells have a massive increase in reactive oxygen species and oxidative stress. And this has actually been known for a long time. Studies by Joan Brugge showed this probably 10 years ago, that anchorage-independent cells have high ROS levels. So, Haifeng did a very interesting experiment. He asked, what is it that tumor cells upregulate acutely to adapt to this oxidative stress? So he cultured NIH3T3 cells in 3D suspension, but wanted to compare vector alone, non-transformed 3T3 cells with 3T3 cells transformed either with mutant KRAS in red or the ETV6 NTRK3 fusion that Paul Sondel mentioned at the beginning. And sure enough, the transformed cells, as expected, survived in these 3D cultures, whereas the non-transformed cells didn't. So what Haifeng did is he asked, what's happening at this very short time period? What's being upregulated under these conditions? And so he wanted to look at this not at the gene expression level, but at the protein level. So the first technique was to look at the acute translatome, what's being synthesized acutely. And so we use a technique called click pulse SILAC, which is an acute translatomic analysis. I'm not going to go into the details, happy to share it later. And he compared that with the total proteome of both cells transformed by mutant KRAS or the NTRK fusion, and looked for commonly upregulated proteins that were not present in the non-transformed cells. And there were actually 31 commonly upregulated proteins that you can see here, details to follow if any are interested in others. But we were particularly interested in these two, ALA1RAP and NRP2, because both are surface proteins and therefore possible immunotherapy targets. So we then, the next step was to ask which of these proteins are potentially upregulated in the types of tumors we study, namely Ewing sarcoma and other sarcomas and pediatric brain tumors. And to our surprise, ALA1RAP was very highly upregulated, or most highly upregulated in Ewing sarcoma. And around this time, our proteomic data also identified ALA1RAP. So, so we've just published this paper collecting all of this data. This is just published a few weeks ago, although it's been online for a couple of weeks ago, or for a couple of months. So maybe I can just take you through some of this data. So first of all, we wanted to confirm that ALA1RAP is a surface expressed protein in Ewing sarcoma, and it is. It's nicely expressed by immunohistochemistry on the cell surface or on cell lines on the cell surface by immunofluorescence. So what is ALA1RAP? So all we knew at this point was that ALA1RAP, otherwise known as ALA1 receptor accessory protein, helps the ALA1 receptor on the surface of myeloid cells to activate myeloid pathways to drive myeloid functions, either through NF-kappa B or the AP1 ATF2 transcription factor complexes. However, while ALA1RAP facilitates this type of signaling in myeloid cells, we failed to find any evidence for IL-1 RAP associated binding to the IL-1 receptor in Ewing sarcoma cells. I won't show you that data as negative data. And there was no evidence for IL-1 RAP dependent IL-1 responsivity in Ewing sarcoma cells. We did see that IL-1 could activate some of these pathways in Ewing sarcoma cells, but this did not have any dependency on IL-1 RAP itself. And actually, we took it a step further. Many of these canonical IL-1 receptor moieties, either IL-1 receptor 1 itself or some of these cofactors, none of them were particularly highly expressed in Ewing sarcoma. So this made us ask the question, what else is going on? What else is IL-1 RAP doing in this setting? So we went back to our faithful proteomics approach, and we did an IL-1 RAP interactome study. And there were lots of interesting proteins that came up, but one that caught our eye is a protein called CD98. And many of you may know that one of the favorite dance partners of CD98 is a protein called XCT, part of the System X cysteine transport system. So basically, cysteine is transported from the extracellular milieu coupled to the export of glutamate. And the cysteine dimer, oxidized dimer is then reduced to cysteine, which is a precursor, as we'll see, for glutathione, which we just talked about. And this is, as I mentioned, the major or a major antioxidant in cells. Now, I just want to mention erastin and sulfasalazine. These are chemical inhibitors of the XCT system, and you'll hear me mention those in a few minutes. So first of all, of course, we had to show that IL-1 RAP is actually interacting with CD98, and we have nice immunofluorescence data to show that. Similarly, for IL-1 RAP and XCT, there's a nice complex formed. And if you don't believe the immunofluorescence, we also did this biochemically using IL-1 RAP pull down. We have some nice antibodies for this. We were able to pull down not only CD98, but also XCT. Interestingly, there seems to be a disulfide bridge between CD98 and IL-1 RAP. That's important for this interaction because we lose the interaction if we mutate that cysteine. We can talk about that later if people are interested. So the other question was, how is IL-1 RAP actually getting activated in Ewing sarcoma cells? And so one of the things we noticed right away is that there was a nice strong correlation between IL-1 RAP mRNA expression and expression of either EWS-FLI1 or EWS-ERG. And sure enough, if we inactivate EWS-FLI1, we have a nice TET-inducible shRNA system in A673 Ewing sarcoma cells where we can titrate the levels of EWS-FLI1 expression. And if we do that and knock down EWS-FLI1, along with that goes a reduction in IL-1 RAP mRNA levels, and that's nicely recapitulated at the protein level. So we really think this is a EWS-FLI1 target. There are a couple of enhancers within or near the IL-1 RAP locus that Haifeng was able to identify, and we see loss of binding with EWS-FLI1 knockdown. And I won't take you through all this in detail, but to make a long story short, when we inactivate EWS-FLI1 expression, then there's a nice loss of active chromatin marks at both of these GGA repeat sites. By the way, Ewing sarcoma fusions bind to GGA microsatellites. So both of these GGA microsatellites bind EWS-FLI1, and we've even put together the beginnings of what we think is a potential BAF-related transcriptional complex containing BAF155 and P300 that we think is underlying the transcriptional activation by EWS-FLI1 at these sites. So we think that IL-1 RAP is a fusion target of both EWS-FLI1 and EWS-ERG. So the story at this point was that as a fusion target, IL-1 RAP is expressed transcriptionally by these targets, enhances the ability of the XCT system to bring in cysteine, which is converted to cystine, which is important for glutathione and ROS mitigation. And fair bit of data that's in the article that you can read about. But basically, if we inactivate IL-1RAP, there's an expected increase in reactive oxygen species in cells. The cells then no longer grow in soft agar colonies, which is a marker of anchorage independent growth, which we can rescue with antioxidants such as N-acetylcysteine. And finally, cells growing in two-dimensional cultures don't seem to care about the presence of IL-1RAP, but they're very dependent on it in these 3D cultures, which have high ROS levels. And finally, we see the expected loss of cystine in cells lacking IL-1RAP, as well as GSH levels. So this all seemed like a very nice, crunchy story. And of course, as usual, these stories are never that simple. And we've made the mistake of doing too many experiments. We were basically just looking for the correlation between loss of IL-1RAP and GSH levels, and how much of that was due to XCT. We figured that if IL-1RAP was ramping up XCT, then if we just blocked XCT in the same cells, that would have the same effect on loss of GSH. And that's kind of what we saw. The levels of GSH with XCT inactivation were kind of the order of IL-1RAP reduction. The surprise was that if we then also knock down IL-1RAP in those cells, so knock down XCT and IL-1RAP, we saw a further reduction of GSH levels. And we confirmed this by chemical inhibition of XCT. What you can see here is that with sulfasalazine or Avastin, which block the XCT system, and that we then knock down IL-1RAP, we get an even further loss of GSH and cystine, as I'll show you in a minute. And these cells were very, very unhappy. So we thought there must be something else going on in the system. And I apologize for this very complicated schematic, but this was horrifying to us as well, because it turns out that if IL-1RAP is helping to get cysteine into the cell and to drive GSH level, it could actually potentially perturb some of these other pathways, including polyamine synthesis, the methionine cycle, de novo synthesis, and something called gamma glupeptide synthesis. So these biochemical metabolic pathways could all be potentially perturbed if we change cystine levels in the cell. And I just want to highlight this de novo cystine synthesis pathway, because we'll come back to it in a minute. So basically, methionine is converted to homocysteine, which then condenses with serine to form cystathionine, which then can be cleaved to form cystine and alpha-ketoglutarate. And we'll come back to that in a minute. But basically, we looked at these pathways and we were kind of horrified, because we thought there's no way that we can sort of systematically go through all these different pathways to see what else might be going on in IL-1RAP knockdown cells. We'd run out of money before we got anywhere. So again, we went back to proteomics, and what Haifeng did is he asked, what are commonly reduced proteins that we can find when we knock down IL-1RAP expression with two different non-overlapping shRNAs. And so we did that, and we found that there, in this quadrant, a number of proteins that were significantly decreased in the absence of IL-1RAP. And one of them really caught our eye. This is a protein called CTH, or cystathionine gamma lyase. This is what I showed you before. Our IL-1 is ramping up XCT, but there's this whole other pathway called the transsulfuration pathway, which makes de novo cystine in cells without needing the XCT system. So we wondered if IL-1RAP was playing a role in that pathway as well. And so to look at that, first of all, what happens if you block XCT with Avastin? Sure enough, the levels of these transsulfuration mRNAs go up. There are two main enzymes, CTH and CBS. IL-1RAP has no influence on the CBS enzyme, either at the mRNA or protein level, but we saw a very nice reduction of CTH mRNA levels and protein levels when we knocked down IL-1RAP. So somehow ALA1RAP is also regulating this CTH enzyme in the transsulfuration pathway. And sure enough, when we went back to Ewing sarcoma data sets, mRNA expression data sets, there's very nice correlations in both sets with high CTH correlating with poor outcome in these diseases. And if we knock down CTH, again in 3D, there's a huge increase in ROS and a loss of soft agar colony formation that we could rescue with N-acetylcysteine. So that was all great, but it turns out, and this was a bit of a struggle in terms of getting the paper published, it turns out that there's a lot of resistance to this transsulfuration pathway as having a role in cancer cells. There's some high-profile groups that have actually actively lobbied against transsulfuration as being important. This pathway, or this paper in I believe this, can't remember the tumor type, but they looked across a number of tumor cells and said that the enzymes for cysteine synthesis or methionine availability are not related to acute sensitivity to loss of cysteine. And then this article by Gina DeNicola actually saying that de novo cysteine synthesis via transsulfuration cannot support intracellular cysteine levels in lung cancer cells. So we really had our work cut out for us to try to prove that this pathway was really important. And so, so we did the following. First of all, it's true that under conditions, non-physiological conditions of very high cysteine levels in the media, then they're right. There's no role for transsulfuration in maintaining cysteine levels, because if you knock down CTH, there's really no difference in cysteine pools as opposed to ALA1RAP. But as you start to lower the extracellular cysteine levels to more physiological levels, then you start to see an effect of CTH reduction. We then did a flux study where we labeled serine cells with C13 serine, and you're all becoming experts at this pathway now. This gets converted to homocysteine, and we were looking to trace the C13 label into GSH. And sure enough, knocking down either ALA1RAP or blocking CTH with a tool compound inhibitor, we completely lost incorporation of C13 into cysteine, and knockdown of CTH itself wiped out the ability of these cells to make cysteine in low cysteine conditions. And these cells were very unhappy. But finally, to really prove the point, and here I've just turned the transsulfuration pathway on its side, I told you that in the methionine cycle, homocysteine is synthesized, and then via these enzymes, cystathionine is converted to cysteine, and then ultimately to glutathione. Haifeng decided to do a really interesting experiment. He wanted to know what happens if you deplete cells of cysteine, the extracellular cysteine, what happens to the cells in the context of homocysteine. And basically, if there's cysteine around, then the cells don't care about this part of the pathway. They can make GSH probably from bringing it in from the extracellular environment. If there's no cysteine in microenvironment, and you block the transsulfuration pathway, then these cells do very poorly. But what Haifeng did is he fed these cells a cell membrane permeable version of homocysteine, and now you can recapitulate the whole pathway. And in this condition, loss of CTH does have a significant impact on the ability of these cells to make GSH. So this really kind of proved the point that this transsulfuration pathway is important. Of course, most of what I've told you about is in vitro, and so we wanted to make sure that these pathways are also relevant in vivo. And so what we do a lot in the lab is implant sarcoma cells under the renal capsule, very well vascularized region, and we get nice tumor growth and metastatic spread in that model. Here you see the blue Ewing sarcoma cells growing from the implanted tumor cells into the adjacent normal kidney. This is completely blocked by blocking ALA1RAP, and this correlates very nicely with a loss of metastatic capacity in the ALA1RAP null cells. And correlates with an increase in oxidative stress in the primary tumors and also loss of GSH expression in these tumors, which I'm not showing you here. What about CTH? Very similar pattern. The parental cells are highly invasive and highly metastatic, but we can dramatically block both features by inactivating CTH. We've tried, I should mention, we've tried to knock out both of these factors. It's extremely difficult in Ewing sarcoma cells because they largely die. Here we're showing a very nice ability of a non-targeting construct of CTH to restore, that's not targeted by the shRNAs, to restore invasive capacity and metastatic capacity. So this is our new model whereby AWA1-RAP, when cysteine levels outside the cell are high, helps to ramp up intake or uptake of cysteine, which is converted to cystine. Yes, and these are activated by gene fusions. But when cysteine levels are low, and somehow ALA1-RAP can switch the system over to using this de novo transsulfuration pathway. So we really see it as kind of a switch between conditions of cysteine replete versus deprived conditions. And you can imagine in an in vivo tumor setting that sometimes there's lots of cysteine around and sometimes not. So cells that are really dependent on glutathione, as Ewing sarcoma cells seem to be, they really need a nimble system to be able to flip back and forth between these different pathways. So there's one more piece to the puzzle that I need to mention to you, and that's the process of ferroptosis. I think the pioneering work of Scott Dixon and Brent Stockwell almost 10 years ago found that under conditions in which there's high ROS load and lots of iron around, lipids get oxidized, and this can reduce the integrity of membranes, and the cells undergo this form of cell death called ferroptosis. This is inhibited by GPX4, which uses glutathione as a cofactor. I told you before that Erastin and sulfasalazine can block XCT, and so these are used as tools to study ferroptosis. And then there's this other iron chelator called ferrostatin that can actually block ferroptosis. And just very simply, any condition that increases ROS production can increase ferroptosis, whereas antioxidant systems such as SLC7, A11, which is XCT, or some of these other factors like NRF2 can actually block lipid peroxidation and block ferroptosis. Looking at the pathway in a little bit more detail, turns out that under conditions of high ROS, either from the mitochondria or ER or other sources, cells drive this reaction called the Fenton reaction, which requires iron. This produces even more reactive oxygen species, which start to impact on membrane lipids, break down the membranes. And as I said, cysteine uptake and glutathione production can help GPX4 to block this pathway. There's been a lot of interest in ferroptosis recently, in that this study from Ken Olive showing that cysteine depletion and ferroptosis is a big parameter in pancreatic ductal adenocarcinoma. And then also recently, a couple of interesting studies suggesting that, first of all, CD8 positive T cells can actually induce ferroptosis in tumor cells. They secrete gamma interferon, which activates this pathway to repress expression of both XCT and SLC3A2, which is CD98. So they actively block ferroptosis resistance in tumor cells, and then macrophages use a different pathway through TGF-beta to also repress expression of XCT. So this begs the question, is ferroptosis resistance a mechanism for immune evasion? And this is something we're very interested in. But just coming back to ALA1-RAP, we showed that in the we use a dye called BODP11 to look for oxidized lipids. And sure enough, if we inactivate ALA1-RAP, there's a huge increase in lipid ROS and ferroptotic death in these cells, Similarly, if we inactivate CTH, there's also a huge increase in lipid loss and ferroptotic cell death. Perhaps best seen here, this is a complicated IncuCyte experiment. If we just look at the purple bars, these are CTH knockdown cells treated with Erastin, so you're blocking both uptake and de novo production. And we can, these cells are doing very poorly, which we can rescue by giving them ferrostatin, this iron chelating agent. So we really think that what's happening is that IL-1RAP has figured out two ways to block ferroptosis. First of all, through cysteine uptake, and then also through de novo cysteine synthesis to produce cystine and GSH. And we think this kind of addresses a long-standing question in the field, which is why is it when you knock down EWS-FLI1 or other protein, oncogenic proteins, do they start to express these high ROS levels? And sure enough, this is a ferroptotic form of cell death, we think, because when we use our DOX system to knock down EWS-FLI1, these cells are very sensitive to Erastin, which you can rescue with ferrostatin. So almost finally, we have also been looking for IL-1RAP expression in other tumor types. This is work we're doing with several groups, including Nader Jabata, where we're looking at high-grade glioma. There's a percentage of high-grade gliomas in pediatric high-grade gliomas that express IL-1RAP, some glioblastomas, pancreatic adenocarcinoma, about 25% express high levels of IL-1RAP, and we're trying to understand the basis of that and looking across other tumor types such as AML, melanoma, and non-Hodgkin's lymphoma. Kind of hot off the press, this is some staining on stage 4 melanoma by immunohistochemistry, so we're pretty excited. These are some slides provided by Alex Losar and MD Anderson. We're pretty excited about that. So then finally, in the last few minutes, I just want to take you through our efforts to target IL-1RAP. This is staining of pediatric normal tissue TMAs, very little expression in pediatric. These are Ewing sarcomas. This is an adult normal tissue TMA. The only place we see it is in placenta. In blood, there's a little bit of expression in myeloid cells and B cells. And so, of course, begs the question, are there antibodies out there we could use therapeutically? And so we teamed up with Mitko Dimitrov and Y. Lee, and they've used their biopanning expertise to come up with some SCFEs that they've converted to fully human antibodies that nicely bind to the surface of cells. These work very well in antibody-dependent cell cytotoxicity assays in our hands and nicely get internalized in Ewing sarcoma cells. And we're in the process of down selecting payloads to generate antibody drug conjugates. We've also generated some with the help, well, actually, with our collaborators, Rimas Arentes and his group at Seattle Children's. He's generated IL-1RAP CARs that seem to work very well in vitro. These are Ewing sarcoma cells expressing CD19, and here's a CD19 CAR. And these IL-1RAP CARs work not as well as a CD19 CAR, but reasonably well in 2 different tumor types to induce in vitro lysis. However, they have not worked as well in vitro, and Rimas and I and others are wondering what's going on. And this is the final data slide here to show that when Rimas and gang used a orthotopic model of Ewing sarcoma in mice, there was a very interesting association between CD11B monocytic cells that collected in the tumor periphery when they injected the mice with activated T cells. And in fact, by immunofluorescence, we see this very interesting phenomenon that T cells appear to be trapped in the tumor periphery, creating this immune excluded phenotype. So we're very interested to know if this is something to do with IL-1RAP itself, or if this is a more general feature for Ewing sarcoma. So stay tuned for that. So just to summarize, what I told you is that Ewing fusions can drive expression of different surface proteins that can block stress responses, and this works in concert with the ability of these fusions to drive cell growth and division. And what I told you is that ALA1RAP and CTH are novel targets in Ewing sarcoma that replenish cystine and GSH pools, either through uptake or transsulfuration. Seems like Ewing sarcoma cells are highly dependent on GSH. And if you block these pathways, these cells are sensitive to ferroptosis, and ALA1RAP is therefore a promising immunotherapeutic target. So I will stop there and let you read some of the people in green, mainly Haifeng Zhang and Alberto Deladelli from my lab, the team at the proteomics team in our center, the CHOP team that has been very very much involved in these studies, Mitko and Y. Lee at U Pittsburgh, and the group at Seattle Children's. So thank you very much for your attention, and I will stop sharing at this point. Sorry if I went a little bit over.
Paul, that was terrific. What a wonderful story, logical, linear, and multifactorial. I want to encourage people to put questions into the Q&A box while we're waiting for them. Let me just ask a first one. It's really intriguing that your IL-1RAP targeted CAR cells don't get into the tumor. There's lots of questions I'm sure that you can be answering. You showed nice data that the Ewing sarcoma that you've expressed CD19 in is susceptible to CD19 CARs. Have you tried that in vivo as well? Do your CD19 cells get to the Ewing sarcoma, or do they not get there?
Yeah, so that's actually going on right now. We were a little bit surprised, and if Rimas is on the call, maybe he can answer that question because they're running those experiments, but that's an ongoing experiment. That's exactly what we were thinking. Is this an ALA1RAP CAR specific question? I mean, it's possible that these monocytes are expressing ALA1RAP itself, and so the CAR T cells expressing an antibody against it are actually getting trapped because they're actually binding to the surface target on macrophages. It's all open territory for answering that question.
All right, we've got a question in the question and answer box. Is it known if IL-1RAP is post-translationally modified, either in normal or tumor cells? And does this affect the XCT complex formation? And would this affect anti-IL-1RAP therapeutic antibody development?
Yeah, that's a great question. And that's one of the things we're exploring, because the question is, how is it that IL-1RAP can switch from the XCT system to the CTH system? The expression levels don't change. We've got a little bit of evidence that there may be different complexes formed under different conditions of cysteine availability. So that's got to be a post-translational modification. And so I don't have an answer for you yet, but we're actively pursuing that. It's a really good question.
So Paul, two people put their questions in the chat box. So if you want to switch gears real quick.
Sure. Okay. So the first question is, sulfasalazine a potential drug for treatment, especially considering it's FDA approved? Yeah, I mean, it's used for, I think, inflammatory bowel disease. We haven't tested it in vivo in this setting because I think the potency to block XCT, the XCT system is not great. And so, there are new forms of Erastin or other blockers of the XCT system that are probably more promising. And some of them are in clinical trials. And sulfasalazine makes sense because it's already FDA approved, but we have not tested it in vivo yet. Let's see if there's another question.
So I think our moderator, Paul, can go back to the Q&A box.
There's one in the Q&A box from Jinghong Wang. For IL-1RAP CAR T cells, do these CAR T cells get exhausted, or have they reduced effector functions?
Yeah, another great question. And we have some evidence that they're getting exhausted, but we're trying to find a way to pull them out of the in vivo setting to ask that question, or to do some kind of, imaging mass cytometry to ask that. This is a fairly new finding. So I apologize if we're not further along in this very obvious set of questions, or addressing this obvious set of questions.
So towards the very end of the talk, you mentioned that you are seeing some IL-1RAP expression in high-grade gliomas and melanoma. But in your analyses of many different tumors, it sure looked like Ewing's was the one that stuck out as the one where IL-1RAP was most expressed. Do you have any preliminary evidence that the functions you found for IL-1RAP in Ewing's might translate to the way it might be working, if it is, in gliomas or melanoma?
Yeah, so we're really interested in that. First of all, how is it being expressed? I should tell you one thing, that we've confirmed in NTRK fusion, ETV6 NTRK3 fusion positive tumors, that they are expressing high levels of IL-1RAP, but that's a rare subgroup. We're interested in the high-grade gliomas because there are different molecular subgroups, and we do know, based on and Nada Jabato's help, that these are H3.3 G27 M mutant gliomas. So there's something about that mutation that's driving IL-1RAP expression on the surface. But your question is more around, is IL-1RAP doing the same thing in these cells in terms of cysteine modulation? And we're very close to having an answer for you. It's difficult to get the right models for this. We're trying to work hard with Nada to actually get that type of studies going, and it's the same with, we think that in the NTRK altered tumors, yes, that there's a modulation of cysteine metabolism, but we haven't proven it in these other tumor systems.
Thanks. We got another question. This is from the Greater Ewing's patient community. Hello, Piero. He's asking, he's first thanking you for all of the great research. Then, in targeting IL-1RAP, have you looked at ways to overcome the immunosuppressive tumor microenvironment?
Well, that's our kind of, that's the golden experiment for all of this. One of the ways to do this, of course, is to develop an antibody drug conjugate, which, will not, as long as it can get into the tumor microenvironment, it's not going to be susceptible to T cell inactivation. So that's one of the reasons that we're actively developing these processes in parallel. And so stay tuned. We have some exciting work that's sort of percolating as we speak.
It's probably time for one last question.
Thanks, Alan. So Kishore Polarsity has asked, has said, this is a brilliant presentation. The anti-IL-1RAP antibody appears to not be so effective by itself. Is there room for improving effector functions via FC modification or bispecific targeting?
Yeah, that's a great question. And so we're actively pursuing the idea of a bispecific, maybe hitting, we're trying to figure out what else should we target. Should we bring in T cells, which might have trouble getting into the tumor microenvironment, or should we enhance the activity of the antibody by co-targeting something like the CD98 surface protein? If we could come in with a sort of bi-pronged approach, maybe we can get the antibody drug conjugate, maybe a bispecific antibody drug conjugate is the way to go with this. That's kind of where we're going with this.
So with that, Dr. Sondel, thank you for moderating today's seminar, and Dr. Sorensen, thank you for an excellent presentation. And I encourage everyone to attend our next Moonshot seminar series on December 16th. We have two junior early stage investigators, and they're going to talk about novel technologies to understand gastric and colorectal cancers. So with that, thank you for
thank you for the opportunity.
Thank you very much. And so with that, we'll wrap it up. Bye-bye.